Hello, I'm Kelly Eversole, the Executive Director of the International Wheat Genome Sequencing Consortium, as well as the International Alliance for Phytobiomes Research. And I want to welcome you to a joint webinar series today. We are privileged to have Justin Faris from ARS, USDA-ARS, who will be giving us a presentation about genetic dissection of disease-resistant mechanisms in wheat. And before that, I'd like to give you kind of a brief presentation about both of these two consortia. For those of you who are not familiar, phytobiomes is the concept of really looking at the complex systems of plant-based agriculture. It's a plant in a specific environment and all of the geophysical and biological components that interact with it. And that is all, and as it is influenced by management practices. Our vision is by 2050 that all farmers will have the ability to use predictive and prescriptive analytics based on geophysical and biological conditions for determining the best combination of crops, management practices, and inputs for any given year for that specific field. We have quite a few sponsors, and they range from small to large companies to governmental research institutes, as well as nonprofit research institutes. The International Wheat Genome Sequencing Consortium's vision is to enhance breeding through an increased understanding of the molecular basis of traits and their allelic diversity, and that's right on target with what Justin is going to be presenting to us today. We have a number of sponsors as well that range from technology providers and governmental institutes to small and large wheat breeding companies. And our next IWGSC webinar will be deciphering the genetic basis of cytoplasmic male sterility and fertility restoration in wheat by Joanna Melanick from the University of Western Australia. And you can go ahead and register for that. The registration is open, and that'll be later this month. So on your webinar, you'll have your dashboard. You will see a number of different buttons and panels, and the presentation will be followed by a Q&A session. The webinar will be posted on both the IWGSC and Phytobiomes Alliance YouTube channels in just a few days, and you can subscribe to these channels so you never miss an update of one of our webinars. If you have any questions and you look at your panel and your dashboard, you'll see a questions button. Please put your questions in the Q&A panel. Do not use the chat, as that will not be seen. You can use the chat panel to talk with fellow attendees or with any of the organizers. And you can already download handouts from the presentations in the handout panel. So with this, I'd like to again welcome Justin, who will be giving us a great presentation today, no doubt, about the genetic dissection of disease-resistant mechanisms hijacked by a necrotrophic pathogen of wheat. Justin, thanks for being with us today, and thanks for USDA helping to sponsor this webinar.
Okay, thank you, Kelly. Thank you for that invitation. Let me get my screen up here. Okay, hopefully, okay, we should be good. Oops. Okay. All right. Yeah, thanks again, Kelly, for this invitation, and thanks for everybody for joining it. Basically, what I want to do today is tell you a little bit of a story and give a little bit of history about the work that we've done to dissect the genetics of this particular host pathogen system that we work on. And so I'll start with just giving a bit of an outline here. I'll start with providing some background and some introduction to the system, to the wheat Parastagonospora nodorum pathosystem, and the inverse gene for gene interactions that go along with that system, including the genes that we've cloned and the work that we've done to compare structure, expression, and function of those genes, and then using that knowledge that we've developed to come up with a molecular model for what's happening at that level. And finally, I will talk about some resistance breeding strategies, and that will include some caveats, some things to consider when breeding for resistance to this pathogen. So let's start with some background. Well, I think everybody on the call is probably pretty familiar with plant pathogens. You know, we have bacteria, we have fungal pathogens, we have nematodes, we have viruses. Among the fungal pathogens, you know, we can categorize these things based on their lifestyles. At one end of that spectrum, we have the biotrophs that have to feed on living tissue. And at the other end, we have necrotrophs that feed on dead or dying tissue. And some examples of wheat biotrophic pathogens would be those that cause, for example, the powdery mildew disease or the rust diseases, stem rust, leaf rust, stripe rust. Let me get my pointer out here. And as far as the necrotrophs, though, we can further subdivide these into generalists and specialists. The generalists act by a little bit more on brute force. They tend to exude a barrage of cell wall degrading enzymes that tend to kill the host. And one example of that would be the Fusarium head blight pathogen. Specialists, on the other hand, are a little bit more sophisticated and a little bit stealthy. A couple examples of those in wheat would be the tan spot pathogen and the Septoria nodorum blotch pathogen. We work with both of these quite often in my lab right now, but today we'll focus on the work that we've done with Septoria nodorum blotch. So Septoria nodorum blotch is the disease, and it's caused by the pathogen Parastagonospora nodorum, which is in the class Dithydiomycetes. And this pathogen can infect leaves as well as glumes, and when it does infect, it causes cell death or necrosis. And this leads to a loss in photosynthetic capacity, which leads to yield loss. And when infestation is severe, we can get losses up to 50%. The pathogen produces necrotrophic effectors. We used to call these things host selective toxins, but there's really nothing toxic about them because they are effectors. They modulate the host. And these effectors are recognized by dominant genes in the host. And when that recognition occurs, there's programmed cell death that leads to disease. And this usually happens in an inverse gene for gene manner. So if you go back to maybe your introductory plant pathology course, you might remember a man named Harold Floor. And it was Harold Floor who came up with the gene for gene hypothesis. And in that model, when we have a dominant gene for resistance in the host and the corresponding AVR gene in the pathogen, we have an incompatible interaction. And this is because there's recognition now of the pathogen by the host. And this recognition leads to a cascade of events, including the hypersensitive response, which is a form of programmed cell death, along with the oxidative burst, the upregulation of defense response genes, and all those things. And the reason this leads to resistance is because the localized cell death that occurs essentially walls off a biotroph. So as I said before, a biotroph needs living cells. And so when the plant kills itself around the infection points, then that pathogen cannot proceed further. It cannot grow. If that pathogen goes unrecognized, like in these three cases, then it basically can grow unchecked, and that's what leads to susceptibility. Now, in the inverse gene for gene model, in this case, when we have a dominant gene for susceptibility in the host and that corresponding necrotrophic effector, we have recognition, and essentially the same response occurs here as it does here. Now, the difference is that in this case, we have a necrotroph. So it's the biology, the pathogen, that allows it to thrive under these circumstances. So it takes advantage of that programmed cell death that occurs, and it grows and it can proliferate, reproduce, and that's what leads to disease. So if this pathogen is not recognized, which is in these three cases, then we have a resistant response. So a necrotroph relies on being recognized by the host, whereas a biotroph tries to evade recognition. And so these necrotrophs have essentially acquired the ability to hijack these mechanisms that we usually find associated with resistance to biotrophs. So these are the interactions involved in this system. Along the left here, I have the wheat genes that we know about, and on the right are the effectors. Most of these are, well, in fact, they're all small secreted proteins. And I'll say that, all the work, most all of the work and research done on the pathogen side is led by Tim Friesen in his lab, which is just down the hall from me. And then it's my lab that works on the host. So in this system, we've got, TSN1 that recognizes SN-tox A, SNN1 that recognizes tox1. I'll come back to SNN2 in a minute. The SNN3, so we have homologous genes, B1 and D1, that can both recognize SN-tox3. We have SNN4 that can recognize tox4 and SNN5 that can recognize tox5. Now, we used to think that SNN2, 6, and 7 each recognized a unique necrotrophic effector. But the Friesen lab has recently cloned this SN-tox2, and they found that SN-tox2, 6, and 7 are all actually one and the same. It's the same protein. And these three genes, SNN2, 6, and 7, all recognize that particular effector. And actually, we know that in wheat, in the host, there's a lot more genes in addition to SNN2, 6, and 7 that can recognize that particular effector. So this is sort of a unique interaction, and it doesn't really follow the inverse gene for gene model. But I'm not going to talk about that any further today, being we're short on time. So as far as the effectors that the Friesen lab has cloned, he's cloned all of them now, except for SN-tox4. We're not really too concerned about, or interested, I guess I should say, in SN4 tox4, because it appears that that interaction plays a very minor role in the development of disease. And on the host side, we've cloned TSN1, SNN1, the SNN3 genes, and SNN5. And we're also working on SNN2 and think that we may have that one close to being cloned. So for you wheat people out there, if you're a wheat geneticist like me, you kind of like to know the chromosome locations of these genes. So just to show you here real quickly, there's 2 on the A genome, SNN4 and 6. SNN1 is on 1B, SNN5 on 4B, 5B has 2 of them, TSN1, and SNN3B1. And so SNN3B1 and D1, the homologous genes, are on the short arms of 5B and 5D. And then 2D has SNN2 and SNN7. So before I go any further, I want to take just a slide here and describe some of these methods that we use to dissect these interactions. So these effectors are usually can be produced in culture, and a lot of times then we can just take culture filtrates of these isolates and infiltrate them in the wheat leaves to identify necrosis-inducing proteins. Or the other thing we can do is wait till the Friesen lab has cloned them, and he'll usually, what they'll do is put them into a yeast expression vector like Pichia pastoris. And then we can express these effectors directly in a little bit more of a pure form. And then we go ahead and we infiltrate them into wheat leaves using a syringe, a needleless syringe. And then we can come back 3 or 5 days later and score them as either sensitive or insensitive. Of course, a sensitive reaction will show this extensive cell death or necrosis throughout the area of infiltration, whereas an insensitive interaction will show no response. And we might get some mechanical damage here where the syringe was held against the leaf. So ultimately what we want to do here then is to identify a mapping population that segregates for this reaction and score that population, because then that will allow us to essentially map the sensitivity gene on a chromosome relative to molecular markers. But now, just because a genotype is sensitive to the infiltrated effector doesn't necessarily mean, or it doesn't tell us what role that sensitivity plays in the development of disease. So that's the next thing we want to determine. And we can do that by inoculating the population with spores of this effector-producing isolate, and then do statistics on it to determine the role of these genes or of these interactions. And so for example, TSN1, which would be the TSN1 tox A interaction, explains up to about 35% of the variation in disease in this case. All right, so moving on now, we'll talk some about the genes that we've cloned and what they are. So I'm going to start with TSN1. There's a little history to TSN1, so I want to go back to about 1987, and that's when tox A was first identified. And it was first identified by Tomas and Bacchus at Kansas State University, and they found it in culture filtrates of Pyrenophora repentans, which is the tan spot fungus. And about 9 years later, I was actually working on my master's degree at North Dakota State, and we did some work to identify the host sensitivity gene and map it, and we named it TSN1. So at this point, we knew that TSN1 tox A, this interaction, was important in the wheat tan spot system. And since then, there's been a lot of work done on this interaction in tan spot. But about 10 years after this, we found the tox A gene was also in Parastagonospora nodorum, and we found that it was horizontally transferred from nodorum to the tan spot pathogen. And so at this point, we know that this interaction was important in at least 2 systems. Now, just a couple of years ago, Peter Solomon's group, and some work led by Megan McDonald, identified tox A also in the wheat spot blotch pathogen. And so basically what that means is that TSN1 serves as a major susceptibility gene for at least 3 economically important wheat pathogens. So of course we wanted to clone TSN1, and we did that, and I guess it's been almost 10 years ago now since we reported that. And so we used map-based cloning, but I'll say, you know, the methods that we used 10 years ago were pretty primitive compared to what we use now. Anyway, this was work that was led by a postdoc in my lab at the time, Hongjing Liu, and my support scientist, Zhenxi Zhang. So TSN1, it turns out, is a gene that has a protein kinase domain, and as well as a nucleotide binding and leucine-rich repeat domains. And if you're familiar with your disease resistance genes, you'll know that we refer to this as an NLR gene. Most biotroph resistance genes are NLR genes, and they don't have this protein kinase domain. So this was a little bit unique in that this kinase was fused with this NLR part. But anyway, like I said, we published this back in 2010, and so we found TSN1 was a PK-NLR. And a couple of years before this, Tom Wolpert did some work and cloned a different effector sensitivity gene, LOVE1, from Arabidopsis, and he showed that that was also an NLR gene. And about the same time, Jeff Benenson's group also cloned a sensitivity gene, the PC gene from sorghum, which was also an NLR. So we looked at this at the time and thought, all right, 3 of these genes are cloned, they're all NLRs, so this is what we're going to be looking for going forward, is NLR genes as we clone more. So if we move from TSN1 now and talk about SNN1, we used a map-based cloning strategy to clone that gene as well. This was work that was led by a PhD student in my lab at the time, Dongjing Shi. But it turns out that SNN1 was not an NLR. Instead, it was a wall-associated kinase gene, which are characterized by an intracellular protein kinase domain, a transmembrane domain, and then extracellular calcium binding and a galacturonide binding domains. Okay, so we published this. This was about 5 years ago. So at this point, if we compare these two genes, things kind of made sense. So TSN1 being an NLR, we know in plant biotroph systems that recognition of these effectors by NLRs typically activates effector-triggered immunity, right? So, and we did a lot of us and others also did a lot of downstream work to show that indeed an ETI pathway was activated. And so this says that nodorum uses tox-A to subvert TSN1 to activate this ETI pathway, so it can take advantage of the cell death. Now with SNN1, it turns out it's a receptor kinase, and again in plant biotroph systems, recognition of these pathogen-associated molecular patterns, or PAMPs, by receptor kinases typically activates PAMP-triggered immunity pathway. So this means that nodorum is using TOX1 to subvert SNN1 to activate this PTI pathway, and again, take advantage of the resulting necrosis or cell death. So looking at this, it made sense. Okay, so this pathogen, we figured, could hijack either one of these main pathways, ETI or PTI, which are the two main biotroph resistance pathways. So we figured going forward, we're going to be either looking for NLRs or receptor kinases. But as we moved on, I'll talk about the SNN3 genes now, and this is much more recent work, so I'll talk about it in a little bit more detail. Again, the SNN3 genes B1 and D1 are homologous on the short arms of 5B and 5D. We were fortunate to find an Agelop's tauschii accession that was sensitive to TOX3 and had the SNN3 D1 gene, because tauschii is the D-genome progenitor of hexaploid wheat and is a diploid. So that made doing genomics and cloning a little bit simpler in that, using that as a system. So this work was led by, again, my sports scientist, Zhenxi Zhang. And Zhenxi looked at about 17,000 gametes to do high resolution mapping. And she was able to locate SNN3D1 to about a 160 kb segment on the AL878 genome. So AL878 is the reference tauschii genome. The problem with AL878, though, is that it's not sensitive to TOX3, so it doesn't have a functional copy of SNN3. So Zhenxi went ahead and developed a back library of an accession that does have SNN3, TA2377. And she assembled the contag and sequenced it, and it turned out that it's about 200 kb larger than the same corresponding segment in AL878. And that's because it had about a 200 kb insertion containing these genes that we have annotated here. If you look through these genes, there were NLRs, there were receptor kinases, there was even a few that were very similar to TSN1. And Zhenxi looked at these genes and interrogated them to try to validate, but we've quickly learned based on sequencing of our mutants that none of these were our gene. And so to make a long story short, it turns out that this gene right here, this PKMSP gene, was SNN3D1. So what is a PKMSP gene? It's a gene that has a protein kinase domain and also a major sperm protein domain. So the major sperm protein is mainly characterized in nematodes, and it's involved in sperm motility. But they're fairly abundant in plants, but we don't know much about their function. We do know that these MSP domains, they tend to form immunoglobulin folds, And if you're familiar with immunoglobulins, those are essentially membrane-bound antibodies in animals. So that means that these things are probably involved in protein-protein interactions. And so again, like I said, they're abundant in plants, but we don't really know what they do. So we're assuming that this is involved in probably in some protein-protein interaction. And based on our EMS-induced point mutations in this gene, which are indicated by these red arrows, we had 3 independent mutations in the MSP domain, which means that this domain is essential for SNN3 function. And this work was just published about a month ago in the Plant Journal. Once we clone one homologous gene, it should be fairly trivial to get the other one. And so at this point, a PhD student in my lab, Catherine Running, came on board and helped with this project. So basically what we did then is just take the sequence of SNN3D1 and do blast against the sequence of chromosome 5B and pulled out the best hit. And we found a very strong hit with about 94% identity. And here we're calling it SNN3B1, but of course, we need to validate that gene. And we were fortunate that to do this, we could use the wheat tilling resources. And most of you wheat folks on the attending here probably are familiar with the tilling resources described by Krasoliva et al. In 2017. But basically what they did is they sequenced about 1,200 EMS mutants from the hexaploid wheat cadenza and 1,500 mutants from the tetraploid chronos, and characterized these mutants, cataloged them, and made all this information available to the community. And so when we took TOX3 and infiltrated chronos and cadenza, we found that cadenza has SNN3, because you can see here it's sensitive to TOX3. So we went to the genome browser and found our gene, and all these little tick marks here down below the gene are variants that are available in cadenza and chronos. So if you click on one of these like we did here, you can pick out that variant and information about it and the consequence and all those things. And so what we did is we ordered up 13 of these cadenza mutant families. And when we tested them with TOX3 by infiltration, we found that 9 were completely insensitive or segregated per reaction. And so that was pretty good confirmation then that this indeed is our SNN3B1 functional gene. That's not quite the end of the story though, because we previously developed 13 TOX3 insensitive mutants in a wheat line called SUMI3, and we thought, well, we can further validate our candidate gene here by sequencing from these mutants. So we did that. We sequenced the SNN3B1 from the mutants in SUMI3, but found that none of them had mutations within the B1 coding region. And so this was a rather a surprise, because we had done the mapping in SUMI3, and the phenotype, the gene mapped right to where we knew SNN3B1 to map. But we did an allelism test, and where we crossed SUMI3 with BG220, which is a TOX3 differential, and we're pretty sure it has SNN3B1. And among the F2 plants, we found 2 that were insensitive to TOX3. And so that would indicate that these two lines did not contain alleles, and suggested that SUMI3 has a different gene, not SNN3B1. So the next question then was, what is the TOX3 sensitivity gene in SUMI3? Well, because we had all of these mutants, we thought we were in good shape maybe to use mute-chrome-seq technology. And so we contacted Jaroslav Dolazel's lab, and as you all know in the wheat community, Jaroslav is the master chromosome sorter. And so his group sorted chromosome 5B from SUMI3 and from each of these mutants, and did the sequencing and assembly. And then we did the sequence comparisons to identify SNPs within common scaffolds. And we found one that was clearly the gene, and we call that gene SNN3B2. So what was SNN3B2? Well, it's a PK-MSP gene but it only had 77% identity to SNN3B1, and it was located about a megabase distal to SNN3B1. So this is the current situation of SNN3B genes on 5B and 5D. We have B1 and D1, which are the homologous genes, and then B2 now, which is about a megabase distal to B1. And I'll say too that we think there's probably more than this. We have pretty good evidence that there's a B3 gene somewhere on here that we're trying to characterize as well. Okay, so if we go back to this slide and add in SNN3 and see how it fits. So again, we had TSN1, which is NLR, indicating the ETI pathway is hijacked, SNN1, which is a receptor kinase, suggesting the PTI pathway is hijacked. So what about a PK-MSP gene? We don't, as far as I know, there's no PK-MSP genes that are directly involved in conferring resistance to a biotrophic pathogen. But I will say that we do know, that a compatible SNN3 tox3 interaction leads to a typical ETI type of response, right? We get oxidative burst, we get upregulation of the defense response genes, and all those things. So it does seem that this gene, or PK-MSP gene, activates a pathway, a ETI type of pathway. And so I wouldn't be surprised in the future if we maybe find some PK-MSP genes that are involved in conditioning resistance to a biotroph. Okay, so we'll move on from SNN3 and talk about one last gene, SNN5, and just make this brief. So I had a master's student, Sapna Sharma, who was working on the saturation mapping of the SNN5 gene, and she was able to narrow that to about a 1.4 megabase region with about 18 high confidence genes. And at the time that Sapna was working on this, John Richards was a postdoc in Tim Friesen's lab. He was working on the cloning of TOX5, and he found that there was structural similarities between TOX3 and TOX5. And so he asked the obvious question, then could SNN5 be a homolog of SNN3? So he took the SNN3 sequence and aligned it to this region and found two candidates. And we developed markers for those candidates, and they both co-segregated with SNN5. So we had a couple of really good candidate genes here without really doing much. And so Sapna graduated, and Catherine Running took over this project. And one of the first things that Catherine did is she infiltrated Chinese Spring and cadenza, and she found out that both of them were sensitive to TOX5. And so that means they both have a functional copy of SNN5. And this was big because Chinese Spring, of course, is the reference genome, and cadenza, again, with the tilling resources available, we made good use of that. So Cat pulled up on the genome browser, we found these two genes, which were basically in tandem. And again, we have all our variants here, and we ordered up a bunch of those for each gene and tested them with TOX5. And for this one, we found about half of the families that we tested were completely insensitive to TOX5, or they segregated for sensitivity. Whereas this one, all 17 mutant families that we tested were completely sensitive. So no change in the mutants in this gene compared to the wild type. So that pretty much confirmed to us then, okay, that this gene right here was SNN5. And so Cat continues to do further validation. In fact, she's already validated it with more mutants, but continues to characterize this gene. Okay, so now that we've got a number of these genes cloned, we can do some comparisons. Yeah, we can look at structure, but this doesn't really tell us a whole lot, I guess, other than, TSN1 is a much bigger gene than the other three. But if we start looking at functional domains, the one thing that stands out right away is that every one of these genes has a protein kinase domain. And so that would suggest then that, signal transduction through phosphorylation is essential for these genes to be functional. As far as the other domains, there's nothing directly the same, but if we consider what these domains do, the LRR and the galacturonide WAC binding domain and these MSP domains, these all seem to be involved in protein-protein interactions. So these are probably the domains involved in perceiving the effectors, whereas the protein kinase, of course, is involved in the signaling. One thing we like to do ever since we cloned TSN1 is to look at mRNA expression levels of these genes during a 12-hour photo period, and we go in and we collect tissue every 3 hours over a three-day stretch. Because when we first looked at TSN1, we found this downregulation through daylight hours, and then it went up through the night hours, and usually peaked here about subjective dawn. And we've seen essentially that same pattern with SNN1. So these two genes were tightly regulated by light. Now, when we looked at SNN3, we've seen something a little different, where SNN3 would increase through the daylight and into the night, and then basically bottom out at subjective dawn. So essentially, we've got the mirror image with SNN3 compared to SN1 and TSN1, but yet all three are tightly regulated by light. And it gets a little more interesting too when we basically turn the lights off on these genes. When we turn the lights off on TSN1 and SNN1, we basically turn the gene off. mRNA levels just drop down to almost nothing, and that's where they stay. And this provides a good explanation for why the TSN1 tox A and the SNN1 tox 1 interactions are light dependent. Okay, so if we infiltrate plants under dark conditions, we get no necrosis, no programmed cell death. All right, and this is probably because we're essentially, by turning the lights off, we're turning these two genes off, and so there's no perception of the effector. But with SNN3, when we turn the lights off, we get a little different story. We get this massive ramping up of mRNA levels within the first few hours, then a decline, but it still stays fairly high through 48 hours. And this provides a good explanation for why SNN3 tox 3 is not light dependent. Okay, we can put these plants in the dark and infiltrate them, and we still get cell death. And this is probably because the mRNA levels are still high enough that we're getting enough protein to recognize tox 3 and turn on those pathways. Other thing we like to look at is the function of these interactions in disease expression. And this is some work I'll talk about that was done by a PhD student in my lab, Amanda Peters-Haugrup. And this was actually some work that she did as part of her master's degree. So we looked at evaluating expression of disease. This is expression of disease now, not gene expression, conferred by these compatible gene-for-gene interactions. And we had this mapping population that's segregated for these 4 sensitivity genes, TSN1, SNN1, 3, and 5. And then she inoculated the population with pathogen isolates that produced various combinations of the effectors, tox A, tox 1, tox 3, and tox 5. And we went ahead and we used a regression to determine the role of these individual interactions in disease development. So like I showed you on that earlier slide, that's how we quantified these interactions. She also did RNA-seq and RT-qPCR to look at expression of these effectors. So there was a whole lot of data presented in this paper. It was published a couple of years ago in Plant Physiology. And I certainly don't have time to go through all of it, but I want to give just an example of the kind of thing that she looked at. So in this example, she's looking at variable expression of 2 of these interactions, SNN1 tox 1 and SNN5 tox 5. So looking at the expression of these 2 interactions in the presence or absence of a third interaction, TSN1 tox A. And so the relevant chromosomes are 1B, 4B, 5B, because we have SNN1 here, SNN5, and TSN1 on 5B. And so the first isolate that she used, that she looked at, was SN2000. And this isolate produces tox A, tox 1, and tox 5. And what she found was that TSN1, or the TSN1 tox A interaction, played a major role in disease susceptibility, explaining 33% of the variation. SNN5 and SNN1 were also significant, explained 7 and 10% of the variation. Now, the next thing she did is she took SN2000, so this engineered isolate. So the Friesen lab engineered SN2000 by knocking out tox A. So they knocked out the tox A gene, and so now we have essentially the same isolate that produces only tox 1 and tox 5. And so when she inoculated the population with this, we see that significance of TSN1 went away, which we would expect because now there's no tox A present. But the significance of SNN5 and SNN1 jumped from 10 to 34 and from 7 to 30. And I should mention too that both of these isolates essentially cause the same level of disease. And so what's happening here is by knocking out tox A, the isolate overcompensated for that, or compensated by increasing the significance of the SNN5 tox 5 interaction and SNN1 tox 1 interaction. So how is it doing this? Well, another thing Amanda looked at expression of these effectors under inoculation with both of these isolates. So in this example here, she's looking at tox 1 mRNA expression 12 to 96 hours after inoculation. With the brown bars are with the wild type isolate. So here you can see just kind of a low level consistent means of expression, and that led to 7% of the disease variation. But when you took the same isolate and knocked out tox A, we see this significant increase in mRNA expression of tox 1. And so that led to an increased level of disease expression. And so what's happening here then is this pathogen has the ability to compensate for the lack of tox A by increasing the mRNA expression levels of other effectors that it has, and to use that to cause more disease. So just a few takeaways regarding the function of these interactions in disease. There's a lot that I didn't talk about, but one is that the effects of these interactions on disease varies depending on the isolate and the host genotype. So in other words, what genes are in the wheat plant, what genes are in the fungal pathogen. And these effects range from additive to epistatic, and even some of these effects can be masked or even inhibited by others. But we know that regulation of these interaction expression occurs at the level of effector gene transcription. And it may be that the pathogen, it has a set of these effector genes, but it probably expresses mainly just those that have a corresponding host sensitivity gene present. Because it kind of makes sense that the pathogen probably doesn't want to waste a lot of energy expressing effector genes that will not induce host cell death. All right. So with all of that knowledge, then we can start to devise somewhat of a molecular model to figure out what's going on at that level. And so let me start with SNN1. So SNN1, as I mentioned, is a membrane spanning protein with an intracellular kinase and these extracellular domains. Now we know that tox1 is secreted, and we know that it interacts directly with the extracellular portion of SNN1. And when this interaction takes place, we know that there's MAP kinase signaling, very strong. We know that the MAP kinase 3 gene is highly upregulated within about 10 minutes after this. And there's also, of course, the upregulation of the defense response genes, the reactive oxygen species, including other hallmarks like DNA laddering and electrolyte leakage. This all leads to plant cell death, which then leads to what we call necrotrophic effector-triggered susceptibility, or NETs. Now, the TSN1 gene is cytoplasmic, and we know that tox1 is secreted and internalized, where it's probably recognized indirectly. And when it is recognized, again, we have the signaling that leads to all of these things, the defense response, oxidative burst, eventually cell death, and susceptibility. Now these two genes we know a little bit less about. Like I said earlier, they're probably membrane associated, but we need to do some more work to learn more about them. And the effectors we know, TOX3 and TOX5 are secreted. We don't know if they're internalized or not, or how they're recognized. Maybe direct, maybe indirect. But we do know that once they are recognized, there's the same thing happens. There's the signaling and upregulation, all these events that lead to programmed cell death. So from a breeder standpoint, we want to look at this and try to devise a strategy to manipulate this to acquire resistance. And so as a breeder, what I would want to do is knock out or eliminate recognition of these effectors. And so we can do that by using marker-assisted elimination to eliminate these dominant susceptibility genes and replace them with either recessives or nulls, so that there's no recognition. Because if we take out recognition, then we can take out this pathway, and that will lead to a resistant phenotype. So we can use marker-assisted elimination. We can also use gene editing, and we're doing this some with Brian Staskowitz's lab at Berkeley. And Brian, one thing he's done is his group has gone in and turned off the TSN1 gene using the system, and you can see when we do that, we get a pretty resistant response compared to when TSN1 is functional. So there's a few things to consider for breeding. These effector sensitivity genes, it's possible they can also act as resistance genes. R genes for resistance to biotrophic pathogens or pests. And this is not without precedence, because the oat, victor, and sensitivity gene, which is an NLR, confers susceptibility to Victoria blight and also resistance to crown rust. And so we need to be careful when we're breeding, because breeding for resistance to one disease could result in susceptibility to another. And also when breeding and introgressing material from uncharacterized sources, we should be careful, because it's possible then that we could inadvertently incorporate a susceptibility gene. So maybe we're breeding for a resistance to rust and we get a nice rust resistant germplasm, but during the process we also incorporated PSN1 or other susceptibility genes, so now we have a highly susceptible germplasm, susceptible to a necrotroph. So in summary, these necrotrophic specialists like nodorum, they actually somewhat trick their host into inducing cell death through recognition of these effectors by sensitivity genes. And the cloning of 4 of these genes, actually we've got more than that clone now. It reveals at least 3 classes. I didn't talk about the tan spot system, but we think we've got another 1 or 2 cloned in that system that actually represents a 4th class. So the point is, though, that these pathogens can target diverse host genes to hijack these pathways. Things like host gene composition, light-regulated expression, epistasis, and additive effects among these interactions, effector gene regulation, all these things contribute to disease expression. So it's not just as simple as a gene for gene or inverse gene for gene interaction. There's a lot of other variables that come into play when it comes right down to disease expression. And resistance can be obtained through marker-assisted elimination of these genes or by gene editing. So just to give a few acknowledgements here at the end, most of this work I'll say was funded by the USDA NIFA Competitive Grants Program. This is a list of people who has worked on these projects in my lab over the past several years. As I said, we've worked closely with the Tim Friesen lab and the folks in his lab. Also the Stephen Shu lab, the Shunwen Lu lab, all these are located at USDA at Fargo. And these are some other collaborators that also assisted in a lot of the work that I talked about today. So with that, I will stop and try to answer any questions.
Thank you very much, Justin. What a really fascinating story, and really highlights the work that you've been doing for the past few years. We have a number of questions, and just to remind everyone, please do put your questions in the Q&A panel. We'll try to get to as many of them as possible, and if not, if we don't get to them during the webinar, we will try to get answers to you after the webinar. The first question is that it appears Chinese spring and cadenza have several SNN genes. Does this make these cultivars more susceptible to stago?
Yes, typically these interactions are additive. You know, their effects on disease are additive. You know, like I say, sometimes they can be epistatic or mask each other depending on what's all involved. But typically, if a genotype has more than one sensitivity gene, they are more susceptible to nodorum because those genes are additive. So this is a little bit different than what you would find in a biotroph system where resistance genes, you know, typically if you have one resistance gene, then that plant is resistant. There's really not too much of an additive effect, at least not in the gene for gene model. But in this case, yes, they're typically additive. And when we've had some genotypes with a lot of these genes present, and they're super susceptible to nodorum. So it's important that from a breeding standpoint that all of these genes be eliminated to obtain resistance.
All right, great. The next question is, are there circadian rhythms in the secretion of toxins during infections?
That's a little harder to answer. The answer is I don't think so. One thing I'll say is that these host genes are, you know, their expression is light regulated, but it's not circadian regulated because, you know, by definition, circadian rhythms, they should be able to run on, you know, after the organism is trained, they should be able to run on for at least 48 hours after you turn the lights off. In this case, we don't see that. We turn the lights off and they just basically the genes turn off. Now, in the pathogen, I don't know. I don't believe anybody has checked whether or not circadian rhythms affect expression of the effectors. The answer is I don't think so.
Okay. In the genetic dissection of host gene interaction slide, could you give a few more details about the inoculate mapping population experiment and the analysis and the QTL analysis?
Okay, let me go back. The person said the genetic dissection methods slide?
Of host gene necrotrophic interaction slide. I don't see a slide number, so. Oh yeah, that's probably it. Yeah.
I'm guessing it's this one. Let's see. So yeah, basically, what we do is we infiltrate these plants. And let me just say, if I don't answer your question, please feel free to email me later and we can discuss further. But so we infiltrate these plants and we get a score or we get a score for whether or not they're sensitive or insensitive. And typically at that level, we can map these things as a Mendelian trait, right? So either it's basically a plus minus. And so we can map that as a sensitivity gene on these genetic linkage maps. So we know right where the gene that confers this reaction to this infiltration, we know where it maps. And so, but at that point, like I said, just because a genotype is sensitive doesn't necessarily mean that it's involved in susceptibility to the fungus. And so the difference is here we're directly infiltrating a purified or a partially purified protein, but that doesn't always respond the same as when you actually take spores and inoculate the plants themselves. So this is the scale of resistant to susceptible when we inoculate plants and score them for disease. We score them on a 0 to 5 scale. 5 is super susceptible, 0 is resistant. So we want to do this. Take the isolate that produces this effector and inoculate the population, and then we can do QTL analysis or regression analysis. And if we get a significant QTL where that effector sensitivity gene maps, then we know that that interaction or that sensitivity gene is involved in conferring disease susceptibility, and to what extent. So hopefully that answers it a little bit. But like I say, if it doesn't, they're welcome to email me about that.
All right, great. The next question is tox A the only suspected case of horizontal gene transfer in these systems?
I believe so. I don't recall any suspected cases of the other genes being transferred. Yeah, I think tox A is the only one that we know about to date.
All right. The next question is, I am working with the biotrophic pathogen causing powdery mildew and found it quite striking that TSN1 has the typical NLR structure of resistance genes. Do you know of any disease that TSN1 causes resistance to?
Right. That's a question we get often, and we've looked for that a little bit, and we haven't found those cases. And I mean, because that could be the case. We guess with not only TSN1 but any of those genes. SNN1 being a wall-associated kinase. You know, wall-associated kinases are also involved in disease resistance to a number of diseases. So that's something that we're on the lookout for. It's not something we have found yet. It may be that these are defeated resistance genes that were just kind of left around or still hanging around in the germplasm. Yeah, the answer is no. We don't know of it as acting as a biotroph resistance gene as of yet, but it's likely that at some point it was in fact that.
All right. The next question is, knocking out the TSN1 and I can barely see this, SNN1 genes, does it cause any other changes in the host beside the possible greater resistance to the host?
Right. The answer is no. We haven't found a phenotype for these knockouts other than conferring resistant. And there's plenty of varieties and cultivars that are lacking these genes that perform very well. So we don't see where they have other functions. At least we haven't been able to ascribe other functions to them. Right, good question.
So do you have any ideas or evidence of what these genes may be involved in besides susceptibility that keeps them in the plants from an evolutionary standpoint?
Right. Yeah, that kind of goes along with the last question. That's another thing we've always kind of wondered is why these things are kept hanging around. They must have some sort of benefit to the plant, but we haven't seen it so far, and it seems like we don't get a detrimental effect when we knock them out. So yeah, the answer is we're not sure why they're. In some cases, there's a lot of areas though where they're not very prevalent. It seems like in some breeding programs they're sort of hanging around a little bit more frequently than in others, and so if they do have a phenotype, it seems that some breeding programs have been able to, kick them out and are able to compensate for it with other genes.
So the next question relates to the interactions within the phyloplane. Would you expect a similar number and complexity of interactions amongst some compatible or incompatible members of the phyloplane microbiome?
Yeah, I'm not sure how much I can comment on that. That's something that we haven't looked at. I guess, but the answer I would say is it's likely because the phytobiome. You go out into a wheat field and there's many pathogens working on a wheat plant at the same time. Biotrophs, necrotrophs, intermediates, and I think it's pretty likely. And I can't. This is just speculation. I have no data to back this up, but I think it's likely that there are interactions where necrotrophs probably affect the ability of biotrophs to affect plants, and vice versa, possibly. Yeah, I think that makes sense, but I have no data or no experimental evidence in that regard.
Great. Well, we have other questions, but we won't be able to get to those today, but we will answer those in writing. But I have one last question for you, Justin. Given the breadth of what's been going on in the past few years with regards to these activities, both in terms of working with host pathogen interactions, what do you see as the major roadblock in the next couple of years that might be removed and allow more rapid advancement in both the understanding and in the development of more resistant varieties?
Well, for the latter, I would say, the freeing up use globally of gene editing technology will be huge. That would eliminate a lot of the breeding headaches. I think as we clone these genes, if a plant breeder develops a superior variety, and then we have the technology to go into those varieties and see what sensitivity or what susceptibility genes it has, and then just knock them out through gene editing, I think that would make the development of superior varieties more efficient. Yeah, I think for the former, unlocking the knowledge, more genome sequences. So it's amazing how these and the tilling resources, all these genomic resources. It's amazing how much more efficient we are today than we were even just 5 to 10 years ago in cloning these genes. If you've seen in my presentation, we've cloned genes without even having to do high resolution mapping, which is a step that used to take years to do. And now with the genome sequences and with the tilling resources, it's a pretty rapid endeavor. And I think more of those resources will really unlock the potential to being able to establish knowledge about these pathways and all the genes involved.
Great. Well, thank you very much for your time today, Justin. And again, it's a great presentation, and it's really fascinating to see the kind of progress that you have made in the past few years. So I want to thank everyone for participating today. And just to remind you, the webinar will be found on the YouTube channels, both the IWGSC channel as well as the Phytobiomes Alliance. So we look forward to seeing you at our next webinar, and hope that all of you will stay safe and enjoy the spring. Thank you.
Thanks, everybody.